Welcome to The Explainer. So today we're exploring a concept that honestly sounds like pure science fiction, but it's rapidly becoming science fact. I want you to just look at the screen for a moment and imagine a future with the next generation of artificial intelligence. Literally, the most powerful computers on Earth are not built on silicon or copper or glass. Instead, what if they're built on living, breathing cells?
For decades, we've kind of just had this running assumption that the human brain is essentially a massive collection of very simple on and off switches. We figured, hey, if we just network enough of these simple switches together, we'll eventually recreate human intelligence, right? But what if we've been fundamentally underestimating the hardware inside our own heads? Because we are on the verge of a massive, absolute paradigm shift in how we understand computation.
Okay, let's dive into this roadmap. We'll be starting microscopic with a single neuron, then we'll scale up to the new field of organoid intelligence, compare silicon to biology, look at real-world wetware in action, and finally, we'll tackle the heavy ethical dilemmas. Starting with Section 1, the ultimate microchip, the hidden power of a single neuron. To really understand where biocomputing is going, we first have to rethink the most basic building block of the brain. Recently, there's been some groundbreaking research out of the Hebrew University of Jerusalem that fundamentally changes exactly how we view individual human brain cells. Professor Idan Segev, who actually led this research, put it perfectly. He said, "What we show is that a single human neuron is itself an extraordinarily sophisticated computing device." For the longest time, the scientific community really just looked at the sheer size and connectivity of the brain, we're talking nearly 100 billion neurons, to explain our intelligence. But Segev and his team discovered that the individual cortical neuron, it's a tiny computational powerhouse all on its own. So the researchers wanted to measure exactly how much computing power a single cell actually had. To do this, they used advanced computer modeling and AI to create an artificial twin of a biological neuron, and they found something absolutely astonishing. To imitate the behavior of just one biological neuron, they couldn't just use a simple artificial node. No, they needed an entire, advanced, deep neural network just to match one single cell. The complexity required to replicate one cell's processing power was completely off the charts.
And you might be wondering, why are they so powerful? Well, it really comes down to their physical structure. Human cortical neurons have these richly branching dendritic trees and highly distinctive electrical characteristics. This unique, intricate structure allows them to perform incredibly sophisticated computations on incoming information right there at the cellular level. They aren't just blindly passing a signal along. They're processing complex visual input, like distinguishing between an image of a cat and a dog within a single cell. This literally alters the entire foundation of how we might build brain-inspired AI.
Which brings us right to Section 2. What is organoid intelligence? Building biological hardware. Because if a single cell is that powerful, what happens when scientists start harnessing clusters of them? How do we actually build with biology? This is where organoid intelligence, or OI, comes in. It's an emerging field that's developing biological computing using 3D cultures of human brain cells, often called cerebral organoids or mini-brains, and then connecting them directly to brain-machine interfaces. We're talking about literally growing a nervous file system in a lab dish to serve as a totally new kind of computing hardware.
Now, what's really interesting about this slide is the term itself, wetware. You know hardware, right? You know software. Well, welcome to wetware. This is the concept of building computer systems using living biological tissue, like brain cells, rather than traditional inorganic materials like silicon and copper. It's a radical, massive departure from the whole shebang that has driven the tech industry for the last 70 years. Let's move into Section 3, silicon versus biology, the limits of traditional AI. We really need to explore why scientists are even attempting this in the first place. It comes down to a clash between our current, incredibly power-hungry machine learning systems and the astonishing efficiency of human biology. And this brilliantly illustrates the stark divide between these two worlds. AI, as we know it today, relies entirely on traditional machine learning. That means massive data centers, huge amounts of electricity, and completely structured information. Organoids, however, use biological learning. When you compare them side by side, the contrast is just staggering. According to researchers like Thomas Hartung at Johns Hopkins, sure, silicon chips might be faster at crunching simple numbers, but they consume massive amounts of energy and require huge, absolutely perfect data sets to learn anything. Brains, on the other hand, they run on roughly the energy of a dim light bulb, and they completely excel at complex parallel processing, even when the data is uncertain or totally incomplete.
Because we are honestly hitting the physical limits of transistor density on computer chips, the push toward AI biology is accelerating fast. Recent reports highlight how new digital organisms and advanced modeling are absolutely essential right now to speed up biological computing. If we literally can't make silicon chips much smaller, we have to look to a totally different medium to continue scaling intelligence.
So this all sounds great in theory, but is anyone actually doing it? Let's transition to Section 4, Brainaware in Action, Testing the Nervous File System. This is where theory meets reality in the lab.
A recent study successfully demonstrated a computing framework they termed Brainaware. But how exactly do you get a lump of cells to compute? The methodology is fascinating. Step 1. Researchers interface with the brain organoid using high-density multi-electrode arrays to send and receive information. Step 2. They apply spatiotemporal electrical stimulation, which, is basically just a fancy way of saying they zap the cells with specific rhythmic electrical pulses. Step 3 is where the magic happens. Through unsupervised learning, the organoid physically reshapes its own functional connectivity in response to that data. It literally rewires itself to process the information.
Let's move to and see how this builds into real-world applications. Brainaware has already achieved some incredible milestones. Researchers have used it to successfully perform speech recognition tasks and even predict complex nonlinear equations. On top of that, bioinformatics platforms like NeuroPlatform have been developed, allowing researchers to experiment with brain organoids remotely via an API. Just think about that for a second. You could literally write code that sends data to a biological brain sitting in another location and receive the computed results back over the cloud. That is just mind-blowing.
But of course, engineering intelligence out of human tissue brings us to our final section, Section 5, the ethical dilemma, the cost of biological computing. As we push the boundaries of wetware, we have to confront the profound societal and moral implications of what we are building.
So, the crucial point is that this technology forces us to ask questions we frankly never faced before. The scientific community is actively debating several core ethical concerns right now. First, there's the potential for these organoids to actually develop sentience, the ability to feel or perceive things. Second, as these organoids grow more complex and capable, there's a real risk of them developing actual consciousness over time. And third, there is the incredibly complex question regarding the stem cell donor. What exactly is the relationship between the human who donated the cells and the resulting organoid intelligence system?
It leaves us with this final, incredibly thought-provoking question to ponder as we watch this field evolve. If a biological computer, grown in a lab and plugged into a machine, achieves sentience, what rights does it have? And what rights does its human donor have over its creations? We are standing on the edge of a frontier that blends biology and technology in ways we're only just beginning to understand. Thank you so much for joining me for this explainer, and definitely keep your eyes on this space, because the future of computing is quite literally alive.